Welcome everyone. Today we're going to be going over the Chapter 7 flipped activities, and this is a discussion in genetics. I'm going to go through this activity fairly quickly. If you have any questions or don't understand something that I have gone over or why that is the correct response, if I don't explain it thoroughly enough, please be sure to send me an email or put a comment down below if you have any questions. So without further ado, alright, so these are the animations, and as always, I'll have them linked in the description box below that you will need to view in order to really understand what's going on here. So, the very first set of questions here are animations. So, pause, go to this website, watch the animation, and then come back. Alright, welcome back. So, the very first question on our activity asks, what are the products of semi-conservative replication for double-stranded DNA molecules? So with the double-stranded DNA molecules, what happens is the strands are going to rip apart from one another, and then they are the template strands, and then you will make um, a corresponding strand off of that, a complementary strand off of that template. And as you know, in DNA, A always pairs with T, or adenine to thymine, guanine to cytosine. So what you end up with is letter C, two double-stranded DNA molecules, each consisting of one paternal strand and one daughter strand. So this, and I'm just going to put little highlighters. So C is our correct answer for that one. Part B, why is DNA replication essential for the cell? Well, in order for life to continue, then, and that's the whole reason for sexual reproduction, right? That's the whole reason for any type of reproduction is to make sure that your genes go into the next generation. So, an organism must copy its DNA to pass genetic information off to its offspring, because if you just kept it all to yourself, then our species would have died eons ago. Next one here. What is the function of the paternal DNA in replication? So remember I talked about pretty briefly here, it's that you have this DNA double-stranded helix that will unwind, and I want you to consider there is a segment in your book, a little chart in your book that talks about all of the enzymes that are required for DNA replication, and one of those in there like helicase and gyrases that help with the winding and uncoiling. So there are enzymes. I want you to keep that in mind, and as you've read the chapter and as you listen to the lecture, that there are enzymes that help to facilitate this process. It doesn't just do it all willy-nilly, but there are specialized proteins that help every step along the way. So when that DNA is unwound, we end up with two of the template strands, or what we consider the paternal DNA, and then the adenines, thymines, guanines, and cytosines that the microorganism picks up or has from its diet, or who knows, that's a whole other story on where that comes from, but it will then make another strand on there. It would make a complementary strand. So what that paternal DNA serves is it serves as a template for DNA replication. Part A, which of the following is DNA strand complementary to CGA, ATC, AGC? And remember, this is just straightforward A to T, C to G sort of business. So C's partner here, and let's pull a pen out because it'll just be easier to write with a pen. So C's partner is G, G's partner is C, A's partner is T. Remember, we're still in DNA, so there's not going to be any fancy U stuff happening yet. A's partner is T, T's partner is A, C's partner is G, A's partner is T, G's partner is C, and C's partner is G. And once again, please forgive my very cryptic handwriting. It is not easy writing with a mouse, at least not for me. It doesn't work very well most of the time. It looks like crap. So the corresponding answer for that is letter B. So moving on, there is another animation for you to watch, and this is the animation over transcription. Remember, for protein synthesis, it's an integrated process that consists of both transcription and translation. In eukaryotic cells, transcription, that means those cells that have a nucleus, transcription is going to happen in the nucleus of the cell, and that's where we make our messenger RNA. And transcription goes from the language of the DNA, the A's and T's and C's and G's, to the language of the RNA, which is A's and U's and C's and G's. And then following transcription, we have this process of translation, where we take this nucleic acid language and then we translate it into an amino acid language. And that's where we make our polypeptide, aka our protein. So for eukaryotic cells, transcription happens in the nucleus, whereas translation is going to happen in the cytoplasm, and that happens with the help and the assistance of ribosomes. In prokaryotic cells, transcription and translation happen simultaneously. That means that, well, prokaryotic cells don't have a nucleus. But what that means is that as the messenger RNA is being transcribed, you can also make that polypeptide sequence. We can go from that language of nucleic acids to that language of amino acids. And make our fully formed protein. So I want you to pause right here and then go watch the animation and come on back. Welcome back. So for part A, transcription produces which of the following? And what we find is that transcription obviously is going to make that messenger RNA because that is the main thing that's making, but it also gives you the transfer RNA and the rRNA that are produced by transcription. And this is a little bit different from what we see in eukaryotic cells because remember, transcription and translation happen simultaneously in prokaryotic cells. So we're going to need this tRNA and rRNA to complete this whole process. Letter B here, according to the animation, which of the following makes messenger RNA from the information stored in the DNA? This is very important. I guarantee you that you will have a question on this on your exam, and it's going to talk about a polymerase. In particular, it's going to talk about DNA polymerase and RNA polymerase. DNA polymerase is responsible for making a new for replicating the DNA, and then RNA polymerase, its whole job is to help add on the correct nucleotide sequences based off of the information from the DNA template to make the messenger RNA strand. So I'm going to put like a little star by that because that's like uber important to remember this polymerase. So on that little chart that I was telling you about, that has all these different enzymes that are on there. And I don't want you to waste time memorizing all the enzymes on that chart, but I do want you to be familiar with, oh, okay, these enzymes do various different jobs in this process of DNA replication and also in protein synthesis. But the one enzymes that I do want you to really know what their job are the polymerases on there. That's how important they are. So this would be RNA polymerase instead of DNA polymerase because we're making messenger RNA. If the question had been asking you how do you make a duplicate strand of DNA, then we would have gone with the DNA polymerase. All right, moving on to letter C here. Ribosomes contain which of the following? And the R in the rRNA stands for ribosomal RNA. So letter A is our best answer for that one. Part D. Which of the following is involved in bringing amino acids to the ribosome? So you remember from the animation that you had your messenger RNA strand and your ribosomal RNA kind of held the whole complex together, but it was the transfer or the tRNA that transferred at one end the anticodon, and then at the other end it had the corresponding amino acid that would correspond to the codon on the messenger RNA. I'm going to repeat that again because that is super important. The amino acid that is carried by the transfer RNA on one end has the anticodon, but on the other end it has the amino acid. That amino acid corresponds to the codon found on the messenger RNA. Important. Rewind it if you need to. Write it down, but you need to know that. Students always get all mixed up when they hear anticodon and they start doing their protein synthesis and then they make their amino acid sequence based off of the anticodon. Do not make your polypeptide amino acid sequence off of the anticodon. It's off of the codon, which is found on the messenger RNA. All right, moving on, letter E. Which of the following can be translated into a protein? Well, the message from the messenger RNA, which came from the DNA, is what gets converted into the polypeptide or protein sequence on there because that transfer RNA carries a corresponding amino acid for the codon on the messenger RNA. So which can be translated into a protein? That would be messenger RNA. Part A, which of the following statements concerning transcription is true? So let's just go ahead and read some of these. All types of RNA in the cell are synthesized by transcription, which uses a portion of the DNA as a template for copying. Okay, that sounds pretty accurate. Promoter sequences signal the end of a gene and mark the place where transcription stops. That's the absolute opposite of what happens. Promoter sequences signal the beginning of the gene, not the end of it. So B doesn't work for us. Letter C, during transcriptions, either chromosomes are copied because the starting point of a gene is unknown. That doesn't really work for us at all because we actually do know where the starting point of the gene is. That's where the promoter sequence happens, and that's where the RNA polymerase will attach to that promoter sequence of the DNA in order to make that messenger RNA. So we do know where the starting point of a gene is. So letter C doesn't work for us either. Letter D, transcription is the process whereby identical copies of DNA are made in preparation for cell division. Well, that is DNA replication. That's almost the exact definition of DNA replication. So that does not work. Transcription is a process where we will take the nucleic acid information from the DNA to make a messenger RNA that will later be translated into a protein. So our best answer for this, the only correct answer, is letter A. All right. Which of the following statements concerning, oh, we did that one. What results from the process of translation? And so what we get as the process of translation is a full polypeptide. And there was an animation that you should have watched. So go back and watch that. I got ahead of myself a little bit here. I get so excited about translation. I mean, like, whoo. Hold me back. All right, so pause this and then watch the animation, and then we'll come back and talk about why that is the correct answer. All right, so what we get out of this whole thing, and we've kind of just generically called it a protein or an amino acid sequence, but that's what a polypeptide is. Peptide are made of amino acids. Poly means several of them. So there are these string of amino acids to pump together. That's a polypeptide. It's not technically a fully formed protein yet because we have to let it get folded, and there are disulfide bonds that have to be made. So there's some modification that has to happen with this polypeptide. But for our purposes and what we're doing on an introductory level, polypeptide, protein, amino acid sequence, those are all the results of translation. Part B, how is bacterial translation different from eukaryotic translation? Well, we kind of talked about this already in the very beginning of this discussion, that in bacteria or prokaryotic cells, bacterial transcription and translation can happen pretty much simultaneously. So bacteria can begin translation before the entire transcription process has actually been terminated. So letter B here. According to the animation, ribosomes along the messenger RNA move along the messenger RNA in what direction? And they move in a 5' to 3' direction. And what you probably have noticed in your readings and in our lectures, that anytime we wanted to make a new DNA strand, if it was on the leading strand, not on the lagging strand, well, even the lagging strand works that way, but then we had little fragments that were those Okazaki fragments, but they still moved in a 5' to 3' direction. And what those primes actually stand for, so these little jobbies right here, what those primes actually stand for is that's just going to be the attachment of the phosphate group onto the sugar. So the deoxyribose is that 5-carbon sugar, that pentose molecule, and whether or not that phosphate group is attached to the 5th carbon or the 3rd carbon, that determines so on one side, they're going to be attached to the 5' carbon, the 5th carbon, and on the other side, they're attached to the 3rd carbon. And they kind of run anti-parallel to one another in the DNA. So one strand is going to go 5' to 3', the other strand is going to go 3' to 5'. So when we have any of these polymerase, whether it's RNA polymerase or DNA polymerase, moving along the strand, what is happening is that it's going to move in a 5' to 3' direction. So on that other strand of DNA that we call the lagging strand, which goes in the 3' to 5' direction, the DNA polymerase will still move 5' to 3', but it does it in little short segments that we call Okazaki fragments. All right, so next thing that we're going to do here, I don't think this one had a animation to it. Actually it did. So there's an animation to this. I should have put that probably at the top so that I knew where my animations were. So go ahead and watch this animation and then come on back. Oops, sorry. I did not mean to click that. All right, so welcome back. So for this question here, so in this animation, what we're talking about are operons. So there are two main operons, or I should say two main classes of operons that we have. We have lac operons, or I'm sorry, inducible operons, and then we have um, those. Wow, I just drew a huge blank. Repressible operons. I don't know why that just escaped me. All right, so we have inducible operons and we have repressible operons, and we really know um, our lac operons in microbiology is like the classic example of an inducible operon. So these operons are just segments of genes that can either be turned off if they're already in the default on position, or turned on if they're already in the default on position. Turned off if they're already in the default on position, and turned on if they're in the default off position for the microorganism. And the lac operon is most well understood of these, and that's why we talk about it a lot. So for part A, it asks which of the following genetic elements is transcribed into a single messenger RNA strand. What we're really looking for are those little structural genes. So they could be gene, I'm just going to say generically, X, Y, and Z. That those are the genes that are going to have to be transcribed and translated in order for this particular operon to either be turned on or turned off. And the reason I keep saying either turned on or turned off is it depends on what class this operon falls into. If it is a lac operon, or I'm sorry, an inducible operon, that means that the presence of something has to turn that operon on. It's in the default position of being turned off, so we need to turn it on. If it is a repressible operon, then that means that um, the operons are going to always be transcribed. Those genes are going to always be transcribed. Those proteins are going to always be made unless we repress it and turn it off. So a tryptophan operon is a good example of a repressible operon. So for letter part A here, the structural genes are what are going to be transcribed when we're looking at this operon. So part B, which operons are always transcribed unless they are deactivated? And those are those repressible operons. So repressible operons, they're doing their thing already, and we want to actually deactivate them and stop them from making those genes. And you might be asking yourself, well, Kelly, why on earth would it, a microorganism want to ever stop making proteins? Well, it's energetically expensive to make proteins and not actually use them. So if there isn't a need to break down this molecule or to make these enzymes or to make these proteins, or you have enough of the byproduct that you needed from making these proteins and enzymes, then you want to turn those particular genes off. You don't want them to consistently keep going on unchecked. So it's a way of kind of balancing. So that's kind of why we would want something to be repressed. So then the next one here, which operons are never transcribed unless activated? And those are going to be our inducible operons. And that's right here. All right, part D, according to the animation, where in the DNA strand does a repressor bind? And from that animation, just straight from the animation, so where that repressor is gonna bind, it's gonna bind at the operator. So whether it is, we want it to bind at that operator there because everything downstream from that is going to be the structural genes that we have. So the operator is kind of like what can get turned off, or what can be replaced, turned off, or what can be turned on. All right, so next animation here. So go ahead and pause this and take a look at that animation and come on back. All right, welcome back. So these next series of animations that we go over are going to be ways that we can have genetic diversity in bacteria. So in the last chapter, in chapter 6, we talked about how one cell becomes two and they're virtually clones of one another. And that process is called binary fission. Um, But we recognize that not all microorganisms are exactly the same, that they do have genetic differences. We have different strains, and even if it's the same species, same genus species, there are still going to be some genetic differences between different classes of microbes. And the way that we have this genetic diversity, because these are asexually reproducing organisms reduced through binary fission, is that we can have conjugation, transformation, and transduction. So this very first animation talked about conjugation. This is the one form of gene transfer that requires that the cells have direct cell-to-cell contact. So it's kind of like, not exactly, but kind of analogous to um, animal sex. That in order for you to have sex and to possibly reproduce with another, you need to come in direct contact with that person or with that person's gametes, their eggs or their sperm. So there has to be direct contact in order for that process to take place. Same thing is true with bacterial conjugation. We're going to have these two bacteria be able to come in direct contact with one another, which is different from transformation as well as transduction. So from our animation, bacterial conjugation is often referred to as bacterial sex. Why is this term inaccurate? Well, this term is pretty inaccurate because conjugation doesn't actually result in new offspring, whereas with human sex and sex the way that we traditionally think about it, there is a potential for a new offspring to form. With conjugation, we don't get a new offspring. What we get is what we call horizontal gene transfer, where this bacteria is now going to be genetically changed after this encounter. So this is the type of sex that will genetically change you. Now, you've probably had sex that changed you in a good way or a bad way, but in this type of conjugation, it's not just psychological, it's actually genetic. Part B, according what must occur for bacterial conjugation to take place? In order for this to take place, as we've already established, the cells have to come into contact with one another, which is different from transformation and transduction. Part C, which statement about conjugation is false? Conjugation is a process of bacterial reproduction. We've already established that there aren't any new offspring that come from this. It's not a vertical gene transfer from paternal generation to the next generation, their children and grandchildren type thing generation. It is just altering the genetic diversity within a generation. So genes are crossing what we call horizontally among bacteria within a generation, and not from grandpa bacteria to dad bacteria to you bacteria. Part D, based on the animation, what is transferred during bacterial conjugation? Well, what's actually going to be transferred is that circular plasmid. So it is a plasmid that is transferred. All right, we have another animation here. So let's pause this, check out the animation. This time we're talking about transformation. And welcome back. So, in transformation, this is the process where the bacteria are actually able to pick up genetic material that is in the environment. So, they can pick up this genetic material, incorporate it into their own, and then they can take on some of the traits that are found in the genetic material that they actually picked up. So, this is really cool and really important for a clinical standpoint because, well, it's really cool because it's just like, wow, it can take genetic information from the environment and just incorporate it into its own. But it's really interesting from a clinical standpoint because when your doctor tells you to take all of your antibiotics, and if you don't take all of your antibiotics, you leave some of those cells on there that just weren't killed right away, those cells can pick up the genetic material of the cells that have been killed that could possibly change its genome and make it resistant to antibiotics. So there are lots of clinical applications to this. And why this is important. And it's just also really interesting to see that this is another method of horizontal gene transfer within a bacterial species. So competent cells are cells that C, can take up DNA from their surrounding environment and integrate it into their own. So these are cells that can actually perform transformation. So one thing I want you to keep in mind with conjugation and transformation so far that we've talked about, and even transduction to some extent, not all bacterial cells can do these different modes of gene transfer. So for conjugation, it required that the cells had a sex pilus or it had a pilus structure. Not all bacterial cells have a pilus. For transformation, it requires that the cell be competent. Not all bacterial cells are competent. So even though these are really cool, neat strategies, it does not mean that all bacteria can use all of these strategies all the time in order to be genetically diverse. Next part here. Mice that are injected with only the R strain of Streptococcus pneumoniae did what? Well, when it had that rough strain on it, remember it didn't have the capsule around it, so their own phagocytotic cells were able to kill it. So the mice were able to stay healthy because their immune system can easily get a hold of this particular strain. Now, with the smooth strain on there, what was the characteristic of the smooth strain that allowed it to evade the immune system of the mice? Well, it had a capsule around it. So that's what this very bottom one says that you can barely see. The cells have a capsule. So what that capsule does is it makes it slippery. It makes it difficult for the phagocytotic cells to adhere to it, to hold on to it. So it's unable to be killed by the immune system, but in turn is able to kill the mouse. Part D, what most likely explains the recovery of the S strain cells from the mouse injected with the heat-killed S strain mixed with the live cell cultures on there? And this is that whole competent cells, the R strain, letter C, picked up the S strain DNA, enabling it to be able to produce a capsule. So now these once rough cells now become encapsulated because they were able to pick up the genetic information of those cells that were killed that's just in their environment, and incorporate it into their own. Letter E, which finding is most surprising from Griffith's experiment? And the finding that he found, what was most surprising, is that the S strain cells are isolated from the blood of the infected mice with heat-killed S strains and live R strains. Because remember, he put dead S strain in there, dead capsule bacteria in there. So a dead capsule bacteria shouldn't give you a dead mouse because it's dead. So it's not actually a live strain. But the fact that he was able to cultivate, so it almost looked like they were zombie bacteria, that they rose from the dead. And I guess to some extent they are zombie bacteria because some of their genetic information was now incorporated into the rough strain cells. So this was the most striking finding because we found S strain cells alive, even though he didn't put live S strain cells in there, and there were no live R strain cells because they had been, or very few, I should say, because they had been transformed. All right, so last animation, I do believe, that we have here. So go ahead and click pause, watch animation, come back. All right, so transduction, our final method of horizontal gene transfer. This is going to be another method that does not require direct cell-to-cell contact, and the cells, just like in transformation, don't even have to meet one another or be in the same physical area for a very long time. This is where a bacteriophage, or a specialized type of virus, will infect a bacteria and inevitably take some of the genetic material from that donor bacteria and then deposit it into another bacteria. The interesting thing about phages, or bacteria viruses, is that they can't replicate on their own. They have to use a host cell's machinery in order to do that. So when the phage is trying to replicate, it inevitably will also copy or transfer or pick up some of the bacteria DNA and the host cell, the bacteria is a host that is trying to make new stuff for. So then when that bacteria phage goes to infect another bacteria, it will also inadvertently transfer some of that genetic material from the donor bacteria to the recipient. So part A, what is unique about transduction compared to normal bacteriophage infection? Well, in this process, you're going to actually transfer DNA from the chromosomes of one bacteria cell to another bacteria cell. Whereas before, with normal bacteriophage infection, we just have more viral genetic material that's being made and then re-encapsulated into the phage. Part B, how is generalized transduction different from specialized transduction? So with generalized transduction, the cycle in which it's initiated is the lactic cycle of a virulent bacteriophage, and with specialized transduction, it's going to be initiated during the lysogenic cycle of a temperate bacteriophage. So the cycle in which it is induced is what differs between generalized transduction and specialized transduction, and the type of phage are going to be different. Part C, a transducing phage contains fragments of the host chromosome instead of the viral genome. And there have to be some of the viral genome on there or the virus couldn't survive. So there is some of the viral genome, but overwhelmingly you're going to have some host chromosome that's going to be transmitted to the recipient bacteria via this bacteriophage. When a transducing phage interacts with a new host cell, what happens is that the DNA from the previous host can recombine with the new host chromosome, and now you have a bacteria that is genetically altered as a result of um, this new DNA being introduced to it from the phage. Which of the following is a method of vertical gene transmission? So far we have been talking about horizontal gene transmission with transduction, transformation, and conjugation, where there's just genetic diversity that happened within the population within the same generation. With vertical gene transfer, those are the genes that are going from one generation to the next. So horizontal, within the same generation, vertical gene transfer from one generation to the next. So the only one of these that follows is cell division. That's the only method bacteria have for vertical gene transfer, and that cell division is what we call binary fission. All right. Frederick Griffith did experiments on genetic transformation. Remember, he had the rough and smooth um, strain of the bacteria there, that the Streptococcus pneumoniae, that either killed the mouse or didn't kill the mouse, depending on whether it was capsulated or not. And then you had like this zombie S strain that came out, even though he didn't put any live S strain bacteria into the mouse. He put the rough strain in there, but they picked up the genetic information from those dead S strain bacteria and incorporated into their own. And when he harvested those bacteria out, what he found were all these live S strain bacteria. So that was transformation. So Griffith's experiments on genetic transformation, which of the following statements about his experiments are accurate? And it is D, that Griffith found that DNA released from those dead virulent bacteria entered the live that were once a virulent bacteria, and virulent just means the ability to cause disease, and change them genetically, causing them to now exhibit those virulent phenotypes, and that it's able to actually kill the mouse. All right, so consider the following statement. So there is a little blurb that you would need to read that comes above this, but I didn't read it. I'm not reading it now. Consider the following statement. Bacteria can exchange genetic material through conjugation without the need for any protein structures or control mechanisms. Is this statement true or false? Well, this statement is false because we already established that the bacteria, in order to do conjugation, A, they have to have physical contact with one another, but they also have to have a sex pilus in order for, which is a protein structure, it's a hollow tube, in order for this process to happen. So this is false. So part C, expression of newly acquired genes. DNA sequencing of E. coli colony oscillated from a nozzle that had biofilm of both bacteria shows that conjugation has occurred. However, the transformed strain of E. coli did not express any new proteins. Blast analysis showed that the gene for the L-mitocytogenes protein is incomplete in E. coli. Sequence comparison shows that E. coli has a truncated version of the gene that begins at nucleotide number 54 and continues through nucleotide 1257, meaning that no start codon is present. Why would this interfere with transcription or translation? So remember, it is very important for a start codon to be there because it says, hey, this is where this particular amino acid sequence starts. And if you're missing any one of those amino acids, a couple of things could happen. A, you're not going to get a fully formed protein, so no new proteins can be established, as we saw with this E. coli. B, you're not going to know exactly where to start, so you can start at 3 or 4, and in this case we started at 54. So things that interfere with transcription and translation here, well, we don't have a promoter region for transcription to begin at because we started like way in the middle, and then there's no start codon for translation. So the DNA polymerase does not recognize an L-mitocytogen G sequence. That's not true because DNA polymerase recognizes A's, T's, C's, and G's. And as long as this genetic information, which they all do, have A's, T's, C's, and G's, is going to recognize it. DNA bases are species specific. That is not true. Those A's, T's, C's, and G's that we're talking about in bacteria are also found in you, which are also found in your plant outdoors, which are also found in the mosquitoes and every living organism on this planet. We are all come from those same 4 nucleotides. The only difference between us and the mosquito, or us and the bacteria, are the number of base pairs that we have, A's and T's, C's and G's, and also the arrangement of those A's and T's, C's and G's on our genome. So translational problems. If the inserted sequence had been complete except for missing the last 54 nucleotides and thus the stop signal, would a working protein be possible? Well, it's kind of a yes or no because DNA sequencing after the inserted gene would need to be done to see whether any stop signals would be in the correct reading frame or possible amino acids would be added to the protein. So sometimes if you add amino acids to it, it's not going to work. Sometimes um, if you do add amino acid sequence to it, it still might work. So both of these conditions are actually probable. All right, and so here are these different types of horizontal gene transfers: gaining a single new gene sequence from outside the cell, transformation, infection by a prophage, transduction, uptake and insertion of a transposable element carrying four genes on there. Those are all horizontal gene transfers. Vertical gene transfers going from one generation to the next: mitosis to form daughter cells, binary fission, and two haploid cells fusing to form a diploid cell. This is actually sexual reproduction on here, and we don't find that in bacteria, but we find it in things like fungus or in protists that are sexually reproducing. And then initiation of transcription. Big things I want you to remember here is that RNA polymerase to the promoter sequence on the DNA strand. RNA polymerase begins synthesizing to creating a DNA bubble on there. The terminator DNA sequences loosen the grip on the RNA polymerase and causes the release of the DNA strand. The smaller subunit of ribosomal RNA attaches to the ribosomal binding site on messenger RNA strand. Then the transfer RNA binds to the start codon. The large subunit of ribosomal RNA is recruited, so that's kind of just holds this whole process here together. All right, and that is the end of chapter 7. I hope you are having an absolutely fantastic morning, noon, or night.